It's a real pleasure to be here and see so many familiar faces. As Elizabeth mentioned, I'm a professor at Wellesley College in computer science, and I'm also directing our Media Art and Sciences program, which is a program that is offered by the Computer Science and the Studio Arts related to Rebecca. Some of the students that have done this, that participated in my work are sitting here today. Some of them are no longer students. I would encourage you to talk to them after the talk as well. And I'll just say hi to my PhD advisor, Rob Jacob. It's a pleasure to have you here. So to provide context to the work that I will present today, I'd like to start with this picture of my research lab at Wellesley College. Usually where I show it, people kind of are excited by the large screens, but standing in front of this large screen is a humbling experience. But Wellesley is a liberal arts college for women, and the researchers in the lab are almost all undergraduate students or recently graduated students. As I said, many of them are here today, and the lab is a very multidisciplinary environment. So students come to work in the lab with background in computer science, media art and sciences, psychology, biology, chemistry, education. And this multidisciplinary nature of the lab is really instrumental for the work that we're doing. We couldn't do it otherwise. And being in a liberal arts environment really helped to explore this field that are at the border between different disciplines. So today I'm going to talk about my work, which is at the intersection of human-computer interaction and genomic and biological data sets. And recent development in genomics and in biological engineering promise not only to transform medicine, but also to address many other pressing problems, such as food production and pollution and even long-duration space exploration. But the vast volume of genomic data set and the complexity of the data pose a real challenge turning this data into insight. So machine learning has been very helpful and has an important role in helping scientists to turn data into insights, but so does information visualization and human-computer interaction. And it's also important to highlight that it's not only scientists that are a population of users who seek to make sense and explore genomic and biological data sets, but also some other populations. So internet technology made genomic and biological data sets available for scientists, but also for future scientists. And this could be college students or children, the general public, which could be people who have access to their own genomic data sets through relatively inexpensive genetic testing, and citizen scientists, who are people who have access to their own genomic data sets, who share and contribute the data with the science and with scientists and with the public, as well as continue to investigate their own data and perhaps their family data. And our work in the last years really targeted each of these user populations. So what I'm going to do today, I'm going to start by introducing very briefly some of the projects that target different populations, and then I'm going to talk in more depth about our focus on the general public and citizen scientists that are seeking to explore their own personal genomes. So GenomeSurfer is a tabletop genome browser that allows novice scientists to work together or to work together with a mentor to explore genomic data sets. And using GenomeSurfer, novice scientists were able to locate an area on a chromosome, to learn more what is already known about a particular gene through access to scientific publications, or what is known about the biological function of that particular gene. They were able to retrieve the DNA or the protein sequence of a particular area on the genome. They were able to annotate area on the genome, as well as to compare different sequences and find areas of similarity. So I'm going to play a video. And as I play the video, you'll notice that GenomeSurfer is implemented on a platform that today is a historical artifact, and that will be the first tabletop computer from Microsoft, which was back then called the Microsoft Surface. Can we play the video? Oh, it's up here somewhere. So we see that the user can access additional information, scientific article about a particular area, particular gene. They can align genomic sequences. Great, so just to give you a taste of this functionality, and we deployed this interface for over 5 semesters in an intermediate course in the genetic base of neuroscience at Wellesley College, and then we used it with novice researchers that participated in the IGEM competition, which is the International Genetic Engineering Competition for undergraduate students, and we worked closely with the Boston University team. We also work with future scientists, and here is an exhibit that we developed in collaboration with the Tech Museum of Innovation in San Jose, California. These pictures are actually taken from the Museum of Science in Boston when we got to deploy this exhibit for a day celebrating building with biology. And the goal of this exhibit is to engage children in biological design and engineering. This is based on research from NASA, MIT, and Berkeley, which look at the application of engineered bacteria to support and sustain long-term space missions through biomanufacturing. So what you see here are the children that are engineering bacteria that could be helpful to sustain this research mission. So the kids, for example, could engineer bacteria that consumes soil that is a resource available on Mars and produce water, which is a resource needed by the astronauts on Mars. And we worked closely with scientists, particularly with Doug Densmore Group for Synthetic Biology at Boston University, where we developed MocoPlanner. And MocoPlanner is a collaborative tool for visualizing and for planning molecular cloning. And molecular cloning is a set of experimental methods in biological engineering that are used to assemble DNA and to plan how to replicate that DNA within host organisms. So this is a complicated process that requires multi-step planning. The technology at that time when we developed it, the top technology for planning was multiple pages of spreadsheets with multiple tabs. So we were trying to allow scientists to work together through this complex planning process. And we have a short video as well. And here you can see that we have a window for each of the steps. They can access information, including the sequence itself. And they can create combinatorial biological design, examining the opportunities of combining different biological parts. They design a template, and the algorithm will explore all the possibilities for them. Great. So I just wanted to give you a flavor of our interaction with different audiences within the area of genomics and biological data sets. But for the rest of the talk, I would like to focus on communicating personal genomic information to non-experts, how non-experts experience their own personal genomes. And this is a collaborative work with Oded Nov at New York University, as well as with Madeleine Ball, who is the director of Open Humans. So I like to start with this graph because it describes how the cost of sequencing a human genome really drops in a rate that is much higher than Moore's law. So the gray line will be Moore's law, and the green line here will be the cost of sequencing a human genome. And we see that in 2001, sequencing the human genome was at about 100 million. And today you can sequence your genome for about $1,000. That's obviously contribute to an increased number of people that have direct access to their own genomic data set. So just out of curiosity, we'll do a show of hand. How many of you have access to your own genomic data set through tests such as 23andMe or Ancestry DNA? Okay, great, me too. It's quite a few. And I assume that if I ask this question in 3 years, many of us will have access to their own genomic data sets. And just to give an overview, personal genomics is a field that is concerned with the sequencing, analysis, and interpretation of an individual's genome. And it's an area that receives much attention in the media, mostly because it promises a health revolution of personalized medicine. But the value of genetic testing is still debated. And individuals share the feeling of information overload when trying to make sense of their genome. Even people that use products such as 23andMe are still overwhelmed by the raw data and by the possibilities of understanding this data. The media likes to position genetic testing as the ultimate quantified self experience, but there are some important differences between the traditional quantified self data sets and personal genomic data sets that we will discuss a little later. So I'm going to move on with a very brief tutorial for personal genetics, and then we'll discuss the unique characteristics of personal genomic and why I think it's a very exciting and interesting and important problem for human-computer interaction researchers and practitioners to work on. And we'll talk about several projects that examine the role of different methods and different human-computer interaction interventions for helping people to experience their own genomes. So as I mentioned, this is going to be a very brief tutorial, but if you're interested in reading more, we published recently a paper in the Journal of Foundations and Trends of Human-Computer Interaction, and it has a more comprehensive tutorial for non-biologists and non-experts in the field. So here's the human genome, which is found in all of our cells and consists of 23 pairs of chromosomes. And with the exception of the sex chromosomes in genetic males, which are annotated here with X and Y, chromosomes exist as matched pairs. One copy is inherited from each biological parent. So each chromosome consists of a single densely packed DNA molecule, which is a linear chain of 4 possible bases that we refer to as A, C, T, and G. And we see here in this picture that genes are sections of the DNA molecule that contain information for creating a particular type of protein that in turn has some particular aspect of the cell biology, which could be translated or not to a particular trait or health effect. In general, the human genome has about 20,000 genes. Genetic variants account for the inherited differences between individuals. So here we see 2 sequences, and this individual might have a pair of T and A bases, and this individual have a C and G. So we call this a genetic variant. And some variants may have health and trait effects. In fact, there are databases that contain millions of genetic variants that are known to be associated with health or traits effects. When an individual carries a genetic variant like this one, it could be present in both copies of the gene. Remember, 1 copy from each biological parent. And in that case, it's called a homozygous variant. Or it could be present in only 1 copy of the gene, and in that case, it's called heterozygous variant. And some health conditions and traits are expressed differently in homozygous versus heterozygous variants. So it is important for individuals, when they look at a gene variant, to know whether this is a homozygous or a heterozygous variant. So that was the end of our quick tutorial. But today, individuals with no formal training, or at least not more formal training than you currently have in genetics, can purchase the genomic information by purchasing a direct-to-consumer genetic testing online, simply by asking to be sent a collection kit. They send back a saliva sample. The genome is being sequenced, or areas of the genomes are being sequenced. There is data mining phase, analysis phase, and an interactive report is being sent back to the user, typically as an online application to be explored. So here is an example of direct-to-consumer genetic testing services. This is the 23andMe kit that you could get home. And today I'm focusing on health and traits information. So I'm not going to show all the ancestry information that you can get from your DNA, but you will get an ancestry report. Or you can get a report that look at several known gene variants, whether they are present or absent from your own genome. So in this individual, which is the sample that 23andMe share, they do not have the E4 variant, which tend to be associated with the Alzheimer's disease. So this is the information that they get, and they can ask for more information, which looks a little bit like this. There is still a chance of developing Alzheimer's and a little bit more statistical information. It will go through several known variants. And there is a nice feature in which you can choose two individuals, typically family members or siblings, and then you can look through areas of similarity. So here is area of half identical or completely identical, the dark blue, in respect to a particular property, which is where this little red marker was set. So if you want to see if you share, for example, the E4 variant that is tend to be associated with a higher risk for Alzheimer's with another family relative, you can use this view. And people can also download their own raw data. And after downloading their data, they can use a variety of tools and databases that exist online for ancestry, for analyzing health and traits, or general kind of resources and databases that connect them to the scientific literature. Many of the users that we establish partnership with, we consider them as citizen scientists, and these are people that decided to undergo genetic testing, but to share the results back with the scientific and community and with the public through the Personal Genome Project, or in its current incarnation, it's known as Open Humans. And the Harvard Personal Genome Project is a database that was originated in a research study that was initiated by George Church at the Harvard Medical School, where personal genome volunteers agreed to share their genomic sequences as well as additional information through a process known as open consent, which is a very careful process of consent. They undergo training. They are tested on their knowledge and understanding of the information that they read and consent to. And part of the consent process is also reviewing and understanding their own personal genomic report. So this is the personal genomic report that the Harvard Personal Genome Project give back to its volunteers or citizen scientists. It's called Get Evidence. And here you see a list of variants that are known to be associated with health conditions or with traits. The clinical importance of this variant, and these numbers were, or these values were calculated manually through a database of experts working together. Here is the impact. Is this a pathogenic or likely benign or a uncertain pathogenic variant? The allele frequency and the population, and a summary of information known about that particular variant. We established partnership with the Personal Genome Project and now with Open Humans, and we work with the users closely. The users are early adopters. They tend to have more knowledge, even though they're not expert in genetics, but they tend to have more knowledge of the subject field. And these are users that are also seeking more ways to interact with their genomes and to conduct their own private investigation. So they typically attend a conference every 3 years for all personal genome volunteers where experts come and present recent results, and also opportunity for us to come and engage with them through in-depth interview and different design workshops. So why am I excited about this? Why do we think that personal genomics is important and interesting for human-computer interaction? Well, first, the results are typically delivered online. So there is an aspect of human-computer interaction. This is personal information. In fact, DNA is the most personal information that you could have. It's really an information about what makes you you. But your genome is also not unique to you. It came from your parents, and it is shared with other family members, and only tiny fraction of your genome is unique to you, and that will be a tiny fraction where de novo mutations happened. This is information that is complex, but also sensitive. But at the same time, it's vary in importance. Some information has clinical implications. Some information is just nice to know. And it has multifaceted uncertainty that is embodied in this information. Even if we do know that a particular gene variant is associated with risk for a particular condition, well, what are the contributions of other genes that perhaps will be preventative? What are the contributions of environmental conditions, of changing in lifestyle? So there is lots of uncertainty that is embodied in this information. The information itself, the data set, is pretty stable. DNA sequencing is relatively accurate. It's actually quite accurate, so it's enough to theoretically sample once throughout your life. But the interpretation or scientific understanding of the data is changing over time. So there is a need for long-term engagement with this data. We also have to rethink consent processes for participating in studies, as well as purchasing products that have to do with making our genetic information available online, or supposedly promising security and privacy while holding our genetic data. And there are other ethical concerns that are associated with genetic data and are interesting for our community of researchers and practitioners. So together with Odette Nov and with Madeleine Ball from the Personal Genome Project and from Open Humans, we've been investigating what the role of human-computer interaction methods and interventions could and should be within the area of personal genomics. And last year at CHI, we presented our Journal of Foundations and Trends paper, which is a more comprehensive paper on our work in human-computer interaction for personal genomics. And we introduced a framework of the role that HCI could play in understanding, informing, and empowering users in this area. So next, I would like to go through each of these areas, understanding, informing, and empowering users, and talk a little bit about open research questions and about our work in this area. So starting with understanding users, here are some research questions. These are the research questions that we thought to address, but there are many research questions that are open and exciting in this area. Why do non-experts acquire personal genomic information? What concerns do they have about engaging with this data? How do they learn? How do they make sense from this information? Which tools do they use? How do they organize their findings? How do they share their findings with other family members, with their communities? How do they validate their finding? And what kind of information do they seek to share? And from in-depth interviews that we conducted with Personal Genomes volunteers, which again I mentioned are early adopters and you can consider them expert users, and with a survey that we conducted, we identified 5 information tasks. The first one has to do with reviewing an annotated report, as one user mentioned, just get an overview of genetic makeup, searching the literature, learn about specific SNPs or specific gene variants, sharing information. And sharing information is really interesting because there are different levels of information that users want to share. They might want to share the fact that they did genetic testing, but not necessarily the result. They might want to share result of a particular gene variants. They might want to share ancestry result. They might only want to share with one participant, but sharing with that one participant also mean disclosing information. So if you share with one sibling, you're disclosing information about the other sibling in that family. They're interested in comparing genome, understanding connections within families, and curating information to compile a list of shared chromosome segments, compile a list of shared gene variants. The second area that we explored had to do with informing users. And there are many research questions that are appealing in this area. We focused on 2, which is how can social features enhance and improve online consent forms? Since most of the genetic testing that is direct to consumer, interaction happens online. You never talk to a genetic consultant. You never talk to a healthcare professional, or you don't have to talk to a healthcare professional. The consent is happening online, and it's very different from traditional genetic testing for a particular condition for which you will go to the hospital, you will meet with a genetic consultant that will provide you the information, answer questions, and then you will go and test a particular very well-defined area in your genome. So we were thinking, well, if it's online, can we use social features to make the consent process better in some aspects? And if we use social annotations of consent form, what will be the influence of particular valence on participants' behavior, whether they consent or not, but also on their perception about their own decisions to consent to participate in a study or not. So we designed this new consent form that, if you're familiar with medium.com, use their API. And on the left side, you'll see that there is the usual content of a consent form. But on the right side, there will be annotations that users could contribute anonymously. So they could highlight an area, and they can say, I like that they thought about the data protection and shared it with us. And then people could like these comments as a way to express that they agree with these particular comments. So in order to study how social annotations impact behavior and perception, we used a little deceit in our study. So we told participants that this is a consent form for a study that will ask them to contribute their genetic data. And we told them that they could read the consent form, and then if they consent, a researcher will contact them for the next steps. We really wanted to study the actual behavior, the actual consent, versus I intend to consent, I would consent if I could. So that was kind of the first element of the set. The second element of the set was that we wrote all the comments, and we also liked our own comments. So there were no users here contributing. And that allowed us to control the balance of the comments. In the beginning, we studied a relatively well-balanced body of comments. And then we studied different conditions of a sum of comments that is more positive or a sum of comments that is more negative in its valence. So this was a between-subject study. It actually had two steps. The first one just studied the presence of balanced comments versus a control group that had a traditional online consent form. And we looked at the consent rates, but we also looked at the perception of to what extent do they feel that their own decision to consent or not was informed, and to what extent they're confident in their decision, and to what extent they trust in the organization that offered this test. And we called our study PGHCI, and we published it under our lab. So we did provide them the information of real researchers that are working in the area. And then in the second study, we had three conditions that had to do with manipulating the comments themselves. So we had a more negative set of comments, a balanced set of comments, and a more positive set of comments. So the good news is we found that there was no significant effect on consent rate. So the people that were not exposed to any comments did not have there was no significant effect of being exposed to comments or not being exposed. And that's good news from our perspective, meaning that it doesn't change the way that people's behavior. It doesn't change the way that people decide to proceed. However, we saw that participants that were exposed to social annotation felt that their decision was more informed. And participants that were exposed to positive valence felt less informed than those in the mixed condition. So having some negative comments or things that question the conditions of the consent form was really helpful to make people feel that they are more informed about their decision. Participants that were exposed to the negative valence were marginally, but only marginally, less trusting than participants exposed to the positive condition, which is not surprising, and less engaged participants, and we measure engagement by the time they spent on the consent form. So people that spent less time on the consent form were more likely to consent when they were exposed to positive valence. So people that did not spend time reading it and they saw all positive comments were more likely than consent than other people. Yes? It's positive versus negative. So basically the sum of the semantics of these comments. So the third area of research had to do with empowering users. And here our research objective were to design tools which allow users to explore and make sense of their personal genomic information. We particularly were interested in designing tools that support long-term engagement, because as we mentioned, with genetic information, it's enough to sample it once, to construct the data sets once, but then you would like to engage with it over time, since the knowledge, the scientific body of knowledge about the genome is changing. And we wanted to facilitate not only engagement, but also to allow users to conduct their own personal research. We also wanted to empower users to contextualize and to act upon their genomic information. And by acting upon their genomic information, we mean go talk to a healthcare professional or go seek more information. It's important to say that we did not reinterpret in any way genomic information. So Odette and I are not in the business of analyzing genomic information. So we worked with the data set of the Personal Genome Project, and we built on their genomic report by creating a new front end to that report. So we created a tool called Genomics, and this is the tool called Genomics. It's an online tool. And what it gives you an overview of the set of variants in your report. So these are all genetic variants. The bigger the variant. Oh, that was the video, not yet. Okay. The bigger the bubble, so each bubble represents a variant, and the bigger the variant, the rarer, like the most rare variant is. It's important to know because rare variants are the ones that you should worry about. If it's something that everyone have and they all live with it, then you shouldn't be really worried about it. But if you have something that is red, means pathogenic, and also very small and very big and very rare, then something that you might want to discuss with your healthcare provider. But it was also important for us not only to communicate how rare it is, also to communicate the possible impact. So red stands for pathogenic, blue stands for protective, and gray stands for benign. And also how certain is the scientific evidence regarding that particular variant. So we see that here on the y-axis, it starts with uncertain scientific evidence, and there is a lot in that basket, especially benign. Doesn't mean you shouldn't worry about it, but it means that for now, with the scientific knowledge, you should really focus on the upper right, where you have kind of red, big, well-established, with high clinical effect variant. Okay, so this could be things that are related to particular cancers, or this blue one is protective against the rotavirus, which is also known to be protection against the stomach flu. So it's a nice one to have. So I think what most people shared with us as they were exploring this visualization is that there is actually less there for them to focus on than they thought originally. So it's a nice way to visualize where you should focus your attention. We also provided another page that allowed them to view, and you'll see that in the video, that allowed them to view these different variants as organized by categories. Things that are related to cancer, things that are related to kind of mental health, things that are related to metabolism, all based on the current scientific evidence and being automatically updated as new evidence appears to encourage long-term engagement. When users click on a particular variant, they get to see information here on that particular variant, and they can curate it, so they can save information about the variants that are interested, and we'll see that in the video. Yes?
I'm just curious, why wouldn't you also have a dimension for whether this information is actionable or not?
Sorry?
Whether this factor is modifiable or actionable or not? So I would add a dimension saying, like, because if you have a gene for Huntington disease, it's important and there's high level of evidence, but there's nothing you can do about it. So I would imagine people would be less interested in knowing that.
So we try to provide scientific information rather than healthcare information. So it was really important for us that we're not providing health knowledge or kind of a healthcare plan or what will be the next steps. Unlike 23andMe, we didn't provide here any recommendations for lifestyle changes or to go see your doctor. We provided the scientific knowledge based on scientific papers about that. So what to do versus what not to do is not something that we provided. But at the same kind of a similar answer to that question could be also that maybe there is nothing to do about that particular condition yet, right? Because we mentioned that the scientific knowledge is evolving. So it's important to keep engaged with your data set, keep visiting, because the scientific knowledge is changing. Maybe there is no cure for Alzheimer yet, right? So you would like to continue and engage. So we didn't want to provide this information of here are the things that you can do, here are the things that you should do. But we did want to provide this information. This is a variant that is related to a possible health effect, unlike a trait effect or just ancestry information. And you should keep engaged and keep checking the knowledge about that particular variant. Going back to the colorful slides that introduced the information task, remember that users were also interested in comparing personal genomes, especially across family members. So we created alternative visualizations for allowing them to compare different genomes. And here we see a linear visualization where each little rectangle is a gene variant. And you can see at the top parent 1, parent 2, biological child 1, biological child 2. And you can see all the information that you had before about the possible effect of the variant and the relative size of the variant. So red stands for pathogenic. The purple here, by the way, is standing for variants that have to do with pharma or drug responses, how sensitive you are to particular drugs, which is knowledge that we have. And they could explore shared area. This is difficult. It's a data set that is complex by itself, and it's much more complex when you have to compare multiple genomes. So part of our research question was, what is the right way to visualize this information? So we created alternative visualizations. We created a linear visualization, and in the video we'll see a sunburst visualization and a table visualization and a Venn diagram, which was a favorite one, but could only compare one other person rather than a set of multiple genomes. And the way we studied this tool is by deploying it to our partners, our citizen scientists at Open Humans, and allowing them to explore it with their own data. We only studied people that already published their genomes, so they were available publicly. We did not ask people to upload. We did not allow people to upload their data sets for this particular purpose. Only people that already shared their data were able to participate in the study. And these are people that I mentioned were already experts on their own genomes as far as non-experts could be. So these are people that already engaged with their genomes. So we were interested in applying insight-based evaluation, asking them, what new did you learn from this particular set of tools that you didn't know about your genome so far? So let's see the video, and then I'll show some evaluation results. Yes?
Something I don't understand. Oh.
Sorry, I'll answer after the video. So here we see the gene variants, and you can get information about a particular variant. Then you can arrange them by category, so drug response or cancer or hearing and vision. And you can switch between these views, and you can curate your own information by saving. These are the different ways that you could use to compare your genome. So there was a table, there is a linear, here is the sunburst that we created. And there is also a Venn diagram that allows you to explore. And whenever you click on a variant, the screen turns off. Whenever you click on a variant, you get additional information. So there was a question. Yes?
Question, the child one, let's see.
This child?
Yeah, on the child one, I see that both parents has one of the, on the next group, the red line there, and the child one, no, not that one.
Not that one.
Is this supposed to inherit? Because both parents have it?
This one?
Here, right
Yes.
here. Both parents have it.
Right.
So shouldn't that be?
It could be. I have to look into the biology of this particular one. So this is a real data set.
Right. So does it mean that maybe one of the parents is not a parent? Or?
It could mean different things.
Yeah.
That could be what it means. It could also mean that we made a mistake.
Yeah, something.
Each parent has two genes, so if
Right.
it's red is one of them, not the other, so they can inherit the other half. So that's perfectly reasonable.
Yeah, but if we have a heterozygous variant with both parents, then this could be a homozygous for a variant that is different from their parents. So this is a real data set. It was actually quite rare to find real data sets of people that shared publicly across their family. Right? So we had very few data sets that had biological parents and biological children, and the data was shared publicly through the Open Humans data set. So that was actually cool that we got to work with real data. So we've seen in the video different mechanisms, and I mentioned that evaluation was mostly based on insight-based evaluation. Of course, we also studied usability, but for us, the most interesting is what other insights people could have about their genome, and I'll just read a couple. It was easy to identify what mutations may or may not be harmful. I've been trying to figure this out on my own, but have been confused, and that's related to different visualization. And this one is something that I found very moving. It is great, my ALS gene that is so bad and scary. I'm a carrier, it won't affect me. I guessed that from previous reports, but it was not clear at all. And we made a point to present carrier information in a different way, since carriers are not always affected by that particular variant in the same way that homozygous variant would affect them. So as a summary, we think that personal genomic information present a really unique and exciting challenge for human-computer interaction researchers and practitioners. It's the combination of characteristics that we discussed that call for new human-computer interaction approaches and interventions. But it's also important to address that new privacy concerns and ethical implications of online interaction with genomic data is something that we should all be thinking about. We do have an upcoming book, and it's currently scheduled for fall 19, which is kind of into the future. But it will explore our work and the general view of experiencing personal genomic and how human-centered computing could transform people's interaction with this field. So stay engaged, it's coming. I want to acknowledge the many people that contributed to this work, and some of them are sitting here in the audience. I'll ask them to kind of say hi. Great. And you can talk to them about this work as well as with me, but I'm very happy to answer questions.
Thank you. What a wonderful talk. Thanks.
Thank you, Michael.
I remember there was a time when geneticists loved to go to Iceland because there hadn't been any conquering there. The records were intact. One knew the ancestry. Is that still relevant for this kind of study?
It's actually very relevant. And if we go to our work where we emphasize the certainty of the scientific evidence, some evidence that we put under a certain has to do with studies that were done on white men in Iceland. What do they mean for biracial women in Asia? Well, we don't know, right? So the scientific evidence is uncertain, and studies that were not replicated or studies that were limited to a very unique set of the population tended to end in that basket. Yes?
I'm curious with this chart, the perception of risk among users when they see an equal number of protective and pathogenic or say benign factors.
Yes. So, so we did not study.
Can you repeat the question?
Yes. Sorry, Corey, would you like to repeat the question?
All right. With this chart, when we see an even number of, say, pathogenic and protective circles, how does that, if I'm, a layperson, how do I perceive that as some, risk, I guess, that I would
so
develop?
We did not study or try to communicate actual risk. What we wanted is, so we did ask them, which genetic variants would you go and discuss with your healthcare provider? And the answer was, and probably should be, what is in the upper right corner. So what there is a well-established evidence and has high clinical effect, high health effect. Some things that don't have a high health effect are just due to traits. The color of your eyes is something that we know with high kind of evidence, with certain evidence, but it's not necessarily a concern for your health, so you shouldn't necessarily go and discuss it with your healthcare provider. But if there is a rare variant, that there is a well-established evidence that it is associated with some kinds of cancer, so there it will fall in the basket of high clinical effect and high certainty, and this is something that you would like to go and discuss. So these are the kind of questions that we explore with them. We try to avoid presenting information directly about the risk and directly about the perception of risk.
Well, I wonder, would they think that those two, the red and the blue, cancel each other out in a way?
We did not find evidence that they think, because once you click on them, you get information about them, and it's quite different. They have different names, and they have a different story associated with them. But that's a good question.
Thank you. This is sort of tangential to the study that you did. I was wondering that these kind of visualization tools, if you in fact use the data from the study more as a sort of a big data type of thing, and you did any type of analytics on it across the different users, or was it mostly meant for individual users?
This was meant for individual users, and I wish we could say that the number of individual users that we studied would amount to big data, but it wasn't, because these were users that shared their personal genomic data online, right, already a limiting factor, and responded and engaged with our study. So we had, if I remember correctly, about 71 participants, which is a good number for this particular population, but far from what I would call big data.
Yeah.
So we did use statistics to quantify their understanding and their perceptions, but not across a population.
Got it. And is that something that you plan to focus on in the future? Or?
Comparing populations?
Yeah.
Probably not, but we're focusing now on comparing families, so allowing people to look at data sets of family members.
All right. Thank you.
So you gave some comments then. People, when they played around with your visualizations, they liked it and they found things. But of course, there are almost an infinite number of different visualizations which you could have come up with. I wonder if you could speak a little more about evaluation and when you came up with multiple versions, how you would test them, or whether it was just using your design intuition.
Yes, so for the first visualization that has to do with the bubble chart, we had a published, so it's actually cited here, a published study in which we compared different visualization techniques. And we tried to look at visualization techniques that are familiar for non-scientific audience. And the bubble chart was the one that was most understandable. And we studied with a large number of users, with about 200 users per condition over the mechanical Turk. So we really wanted to study kind of the understanding of the general public of these particular visualizations. The more challenging aspect was coming up with visualizations for comparing multiple genomes. And here we went with tools for scientists. So we explored what visualizations are currently used in the state-of-the-art tools for scientists to compare multiple genomes. And we tried to simplify them. So there are multiple tools that use linear visualizations. There are tools that use this sunburst circular visualization. There are tools that use Venn diagrams. And we tried to start with this expert visualization and simplify it. And again, we went through multiple iterations of only studying understanding rather than insight. So only the usability of the particular visualizations with mechanical Turk users, because we wanted to study their perception and understanding of the general public.
One more quick follow-up. Any more traditional like usability evaluations where you would sit with someone as they use the tool to assess their understanding and confusions?
Always. So this is how we start. We call them kind of pilots. But yes, we studied kind of many people coming to the lab. We sat with them, we discussed, and we iterated. And then we went through multiple mechanical Turk iterations. And only then we deployed that for people with their own data. We tried to reach the point, because also the data set of people that shared publicly their genome is so limited, we really tried that by the time we get to work with this population, we kind of eliminated lots of the usability challenges that come before.
In your other slide with the bubble visualization, that one, yes, are all those different bubbles representing different gene variants that all contribute to the same phenotypical condition, where a red and a blue may potentially cancel out, or are they representing different conditions, different phenotypes?
Different. Each one of them is different. There could be, depends on the person, there could be a data set that includes something that contributes toward a particular cancer and something that contributes towards protecting from that particular cancer. These are particularly challenging because there is no good way so far that we noticed in commercial reports on quantifying what's more. Do you have more of the better protective one? And will that cancel the effect? So when I talked about multi-dimensions and multifacets of uncertainty, part of the uncertainty has to do with you have multiple genes that might be associated with the same condition but contribute to the effect in different ways, protective or pathogenic, and in many cases we don't have good scientific evidence to understand the interaction of these genes. So as I mentioned, we did not present new interpretation of the genetic reports. In the original Get Evidence report of the Harvard Personal Genome Project, they just listed them and described what is the contribution of each particular variant. And that's what we did. I noticed that in 23andMe they do the same. They just list them separately, but they don't talk about the interaction between them, and they definitely not quantify the interaction between them, which one you have more.
So two bubbles there may be part of the same pathway, and they may be completely unrelated.
In this data set, they're unrelated.
They're unrelated.
Yes.
And a related question, when 23andMe provides their data set, are they providing both heterozygous information, and what proportion of the entire 3 billion base pairs of the genome do they provide? It's not whole genome sequencing.
Very small, so they change over time, right? Because they're now being approved to release more and more tests. Mm-hmm. But the last time I looked, it's about a few dozens carrier status and a few dozens kind of genetic variants that they would provide responses to. I actually did my 23andMe reports before the FDA closed, so I did it in 2014. And if I remember correctly, it was something about 150 different gene variants. So it's like a small, discrete number.
And does that include the heterozygous information?
It included both.
It does. Okay, thank you.
But I don't remember the exact number that they have right now.
Have you tried to apply these techniques to any epigenetic modifications and epigenetic data, and also including microbiome data from that project and things like that?
That's a great question, because this is what we would like. Yes. Can you please repeat the question?
The Open Humans project has an associated project called the Open Gut, I believe, which has produced a lot of very interesting data. And I was wondering if you had begun applying techniques to that and also to trying to analyze and explain epigenetic data.
So this is what we're working on now. So this is where we're heading. We're very excited by studying genomes together with microbiome information. And this is where we're heading next. I don't have results to present. We have kind of some particular interaction designs. But I think that so much of our health would be explained in the future by our microbiome, and we would like to be able to communicate that information. It's hard because if the knowledge about our genome is evolving, then the knowledge about microbiome is really at a very early stage. But we're working with Open Genomes, and we're looking at these data sets, and we're thinking that's our next step.
Just to follow up on what you said, so you used this very nice approach of mechanical Turk to evaluate different prototypes. I was just curious what matrix do you use for evaluation for comparing one to another? On what basis did you choose the best one or the final version?
Right. So we use several metrics. Of course, we used a fictional data set. So it was a fictional data set that was composed from multiple real sets that were shared over open humans. And we looked at objective knowledge on subjective understanding. So objective knowledge, there was a questionnaire that our personal genomic expert, Madeleine Ball, developed together with our students. And that was a questionnaire that asked questions, for example, such as which of the variants in which report you would like to discuss with your healthcare provider. Which of the variants of your reports is heterozygous versus homozygous. So we asked specific questions, and we had a set of 12 questions. And then we also asked users subjectively, to what extent do you feel confident in your answers? And it was interesting. Part of our results is, part of our contributions is really indicating a gap between the objective knowledge and the subjective perception of knowledge. People that got it all right were not necessarily confident, and people that were very confident didn't necessarily get it right. And when we chose the bubble diagram, this was the diagram where people got it correctly and were also confident in their understanding.
Going back to the stuff that you talked about at the very beginning about privacy, I'm conscious, given what's happened in the last several months with Facebook and Cambridge Analytica, that sort of thing, that that some people are very concerned about how their personal data might be used in the wrong hands. I can remember a conversation with a healthcare professional about 10 years ago saying if you want to get certain tests done, don't go to your own doctor's office because that'll end up hitting your insurance ultimately if you have something negative there. So just using that type of example, I'm curious to hear how conversations on this topic have evolved in the last several months, given that whole Facebook incident.
So conversations are evolving, and there are two aspects. One is privacy, and the other one is security. So if you did 23andMe testing, your data is sitting on their server, and as long as their server is protected, your data is protected, and if their server is being hacked, then that's a challenge. And that happened with Ancestry, I think it's called, it's not Ancestry DNA, it's... yes. Last week they published that there was a break on their server. They claimed that no DNA data, no genetic data was kind of shared and stole, but only username information that was kept on a separate server from the actual data itself, and that all information was encrypted. But this is an aspect of data security. Privacy and protection against discrimination is a different issue. So there is GINA, which is the Genetic Non-Discrimination Act, that protects against discrimination in health insurance, but it doesn't protect against discrimination in life insurance. And as more people will get their data and will have access, they will have access, and perhaps others will have access to this information, it's something that we as a community have to be aware of. Also recent news was the use of Ancestry DNA websites to look at matching for criminal detection, right? The California case in which I think it was the Golden Gate Raper was identified based on ancestry data that was shared by a very distant relative that we can assume was never thinking that their data would be used in that way. Of course, we don't know what other ways people could use our data. The Cambridge Analytica is an example of a kind of misuse of data that people thought they're sharing for a different purpose. So yes, there are all these issues that we have to consider and be aware of, and it's definitely a developing area of understanding for us. Yes.
I was wondering, you mentioned sort of replication as being one of the criteria, but sort of how did you classify the level of evidence?
So again, we did not classify the level of evidence. We went with the classification that was used by the Harvard Personal Genome.
Oh, okay.
So we did not, we only presented.
Do you know how
data.
they did it?
Mostly by crowdsourcing different experts.
Okay, interesting. Then I guess sort of related to that, I was curious if you had looked at kind of another way of presenting this data by showing kind of how these different bubbles sort of over time as evidence is generated might actually move from category to category. I could sort of imagine that being maybe a source of anxiety, but also perhaps even a source of relief if people kind of see, look, these are sort of, this is based on what we know now, this is not fixed, right? As sort of new evidence emerges, actually our understanding of these things change, but that might be sort of interesting as another way of sort of bringing the user into an understanding of how kind of science evolves.
Yes, so we were tackling this in 3 different ways. We did not have data set that we could visualize where this actually changed, so we only have interaction designs, but not actual visualizations of real data. But in our interaction designs, we showed kind of trajectory, things moved. We also have a notebooks feature that, again, because we didn't have real data for the interaction design, we didn't share it here, but a notebook feature that shows you over time the different pages where your information was when you start curating it and when it is now, and kind of push new scientific evidence for you, a little bit like a Pinterest board. So it kind of pushed your articles that are related to gene variants that you decided to save. And you could also read and save, pin things, right, like Pinterest, into your notebooks as you go, and it will show you on a timeline what you saved when. But we didn't have the scientific knowledge. Development was not as rapid as our interaction design, so we don't have that to show. I think we have a question there as well.
Hi. I wanted to mention that Science News has a 3-part article they're currently publishing all about the various companies that are doing this and what the various properties of.
Oh, great.
So the question I have for this is, were these whole genome, and did it include mitochondrial DNA, or is it just SNP data, or just introns, or what?
This was exome, so it was a 23andMe data set. So we start by interviewing people that had their old genome sequenced, but the people that we gave this tool to were Personal Genomes volunteer that share online the 23andMe data set.
Which I think is just.
It's just
SNPs,
it's just SNPs, and it's kind of a large number of genes, so it's only exome.
Yeah.
I'll follow up on that. I think that basically answered my question. But, it's understood that that's a small subset of the genome that's understood to be significant, and also that there's this. You're only looking at either that subset or even a smaller subset that's provided through 23andMe. So does that explain all the filtering that applies to the graphs and charts that we've seen? So it's a relatively small amount of information.
Yes.
That explains it.
That's explain it,
Okay.
right?
Did you mostly restrict yourself to the subsets of data that were analyzed by 23andMe, or did you also look at the whole genome sequences available in the PGP project?
So when we use visualization, we use the 20 people that shared publicly the 23andMe data. When we interviewed people to learn about their experiences and understand their motivations, we interviewed people that also had access to their whole genome data. But what you see here in the visualization is 23andMe data.
Does genomics also work with the full PGP? Okay.
Yes.
So
we showed it, but the number of people was very small. So we showed it, I think, to 7 people that came to the GET conference and we sat with and had interaction with.
So I have a question. So, so most of the design and usability guidelines that's in the biological visualization community right now are founded on domain expert evaluations. Insight-based evaluation methodology was created with scientists, and I'm wondering if any of the studies that you've done with more novice users has shown any differences in guideline or visualization preferences, or any of the kind of typical, expert versus novice studies that have been done?
So we did not compare expert versus novice directly, right? We didn't ask experts to interpret the same data sets, and we didn't ask novices to use experts' tools. So
Yeah.
I don't have comparative data to share. I'll say that some of the expert tools are not necessarily using particular design guidelines. A lot of the visualization tools that when we interviewed the scientists, they're building in order to solve a particular problem. So they're not necessarily something that they try to generalize, or they aspire even that other scientists besides their research group will be using, but it's really investing all this effort in solving a really important problem that research group is working on. And since you're working with experts, they have tolerance for learning how to use and how to understand the visualization. There are tools that are more generic, especially genome browsers that use different visualizations. When we worked with novice scientists, that will be undergraduate students or beginning master students that are entering and working in research labs, we use some of the standard kind of state-of-the-art tools. But we didn't study particular guidelines. We did kind of start form guidelines for non-expert users, even the use of color. So when you look at gene expressions visualizations, a lot of the experts ones are using red-green scale. So we started with red-green, but red-green not working very well for the general, possibly colorblind population, right? So we developed this new color scheme that has to do with blue to red. And we went through several iterations. I know it looks really similar to the red-green one, but we actually went through different shades and different gradients of the red-blue data sets. And we found that these data sets, after several iterations, is actually working with this color coding.
Quick question, but first, I don't think ever heard so many questions after a talk, which I think is a testament to the quality and interestingness of your talk. 23andMe often forces you to go through a actually small, what they call tutorial, before they even give you some of their reports. Do you have any information or data about whether that's effective and how best to do it? And I mean, there's many questions one could ask about that.
Yes, so we did not study their tutorial. However, when we ran studies on Mechanical Turk, and we ran, I think by now, more than 1,000 users on Mechanical Turk on different visualizations of personal genomic information across the many years, we had to train them, right? Because we couldn't expect that they would start by understanding basic concept of personal genetics. So we developed a tutorial that was based on materials developed by the Personal Genome Project as part of the Open Consent and by the Personal Genetic Education Foundation. And we developed that tutorial and we iterated on that tutorial, and this is how we start all of our Mechanical Turk studies, with this tutorial that have some quiz questions, and people that cannot answer all these questions would not continue to the next stage. However, I have to say that we had a very small number of people that would not continue to the next stage, and I don't think it was because of non-understanding. I think it was probably because as Mechanical Turkers, they realized that this study requires level of attention and commitment that is different from other studies on the Mechanical Turk.
Have you seen any interest from either clinical practitioners or the genetic counseling community in your work so far?
Yes, which is why we're writing the book. So the offer to write the book really came after we presented at CHI a similar presentation, which is more like an overview of our work, which is what is published in the Foundations and Trends of HCI journal paper. And a publisher, the Cambridge Press, which also work with different populations, not only the CHI community, but also the genetic community, came from a genetic conference, and they said, well, that would be great. How can you reach to that community? How about we work together on a book. So this is kind of what led to writing this book, really seeking to reach out to this community. We did have some people in industry that approached us for consulting, which worked out or didn't work out, depends on the circumstances. We're really committed to working with the open science concepts. So we're currently collaborating with the Personal Genomes and continue our partnership, but also with the Human Gut project, which hopefully is kind of where we're going next.